Okay, so the last question we need to deal with is basically why do we use animal models? So we could talk about, the different types of animal models, talking about knockout models where we eliminate all of the expression of a particular gene, or we can talk about transgenic models where either we create an expression that never existed before, or we enhance the expression of something that's already in place. But really, why do we use animal models in general when we know that there are significant differences between the human genome and the genome of various species? Okay, why do we use these animal models, and to what extent can we use them? Yeah. You didn't? Okay. Sorry. Were you going to ask if I'm recording it?
No.
Okay. Were you going to tell me I was doing something wrong? Okay. Now I'm all excited. What was going to happen? Yeah, Mia. Are you going to tell me I'm doing something wrong?
Oh my gosh.
Okay.
Sorry.
No, it's fine.
I was wondering, do you have those slides available on Canvas?
These slides are on Canvas now, aren't they?
Mm-hmm.
They aren't?
It only has the key terms.
It only has the key terms? Oh, that's terrible.
Yeah.
Oh. Yeah. Wow. Is that what you were going to tell me?
No.
So you were going to tell me I was doing something wrong. This is kind of what my life has become, I think. It's not even really like time of a semester anymore. It's just my ability to remember things is it's not good. It's just not good. I make myself notes. David, you can't drink in here. I know, but you still you can't do it. Okay. So now it's on there, and it's on there as studying complex diseases 2-1. Yeah, it's there. Okay, now going back. All of this is in the recording. I'll have to delete all this now. No, I'll just leave it in there. Yeah, I'll keep it in there. Yeah. Okay, so during this little stretch, we're not gonna focus so much on the shortcomings of using animal models, because those are obvious, right? The animal, no matter how similar their genome is to the human genome, they are not humans, right? And so when we're doing these animal models, there's just a reality that they're not going to give us the full picture of what this would look like in humans. However, they are going to give us a much better picture than nothing, which a lot of the times that's the challenge we have, is because there are so many ethical issues with trying to do these genetic studies using human specimens, that it's like you either have nothing or you have something, right? And something is always better than nothing. Well, not always, but something is usually better than nothing. So, some of the benefits. We can take these various strains, we can take these various, groups of animals, and then here, we should probably do these 2 points together. We can achieve reproductive isolation in animal models very easily, okay? Much more easily than we can in humans, right? And so some of the times, cultural differences are enough to create partial reproductive isolation in humans, but no people group, unless they are extremely remote, is reproductively isolated. But we can do that in these model animals. We can develop a strain of mice that are reproductively isolated and become very different genetically than other strains of mice. And then what we can do is cross those strains with another strain, and we can basically get to a point where we're really good at, if you want a mouse with a particular feature, like you want a mouse that's going to develop diabetes without having to just feed it an incredibly awful diet, but it's still going to develop type 2 diabetes, you could do that, and we do that. To do research on type 2 and even on type 1 diabetes as well. If you want a mouse with type 1 diabetes, you can do that. And so what this allows us to do is basically to think up any combination you can imagine. we can probably accomplish a mouse model to represent that, right? You want to study the interface between one complex genetic illness and a second complex genetic illness, you can do that. Where that would be really difficult to accomplish in humans, right? If you wanted to study how does this particular complex genetic illness, let's say type 2 diabetes, and this other one, a specific type of cardiovascular disease, yeah, you may have a subset of humans that have both those genetic illnesses, but you're not going to get a big enough sample to do anything really meaningful, and then on top of that, there are ethical concerns. But you can create mice that have those combinations. Another thing to realize is yes, even though humans and these animals that we're using are very different, in the same way one individual human is very different from another individual human genetically, there are certain aspects of it that are incredibly similar. And this is developmentally, this is genetically, this is physiologically, this is immunologically, right? There are certain ways in which various animals are very, very similar. Very similar. Which is interesting. It's interesting that it works that way. That even though you may have an animal that's less alike humans genetically, but more like humans immunologically, which is just really fascinating. Like pigs, for example. Pigs are very similar to humans physiologically and immunologically, but they're not nearly as similar genetically as some other animals are. And it's like, well, why is that the case? And it's like, I don't know, but it sure makes it convenient to use pigs as a model for how humans would respond to something physiologically or immunologically. Yeah.
Do you think that those other similarities are due to like similar environments?
Possibly, yeah. Possibly, but the domestication of pigs is not as old as the domestication of other animals that aren't as similar to us, immunologically and physiologically.
But like their diet's kind of similar-ish.
Oh, yeah. I mean, pigs eat pigs. We eat pigs, right? Pigs eat pretty much anything you give them, and we eat pretty much anything you give them. I mean, the difference is pigs are caprophagous to the extreme, which means they like to eat feces. And so if you give a pig a taste for feces, it'll follow humans around waiting for them to poop.
Ew. Oh.
And most humans aren't quite like that, but yeah, I mean, pigs are definitely omnivorous. We're omnivorous, right? And so, we domesticated, for instance, we domesticated cattle and goats and sheep before we domesticated pigs. Pigs are fairly recent domestication as far as evidence goes. But you probably wouldn't anticipate us to be as similar because they're entirely herbivorous. Although, actually, you could feed a cow and it would eat it, which is one of the complications with mad cow's disease when you're using cow tissue. Anyways, but that's another story for another time. But yeah, I mean, there is something to that. Sure, yeah, there's definitely something to it, and so we can explain why this might be the case. But really, it doesn't really matter why, but it sure does make it convenient to use some of these animals as really nice models, proxies, if you will, for humans. Avoids many of the ethical concerns. Notice it doesn't say avoids all of the ethical concerns. I mean, the reality is, at least in most of our mammal species, they respond physiologically to pain the way we respond physiologically to pain. Right? And so it's very obvious that they experience pain in a similar way that we experience pain, which creates some ethical issues. And they even respond physiologically to adverse, what's the term I want to look for? Just adverse environments, right? When you put them in things that are particularly stressful, they respond physiologically the way a human would if you put them in an environment that's particularly stressful. That's not actual physical pain, but it's emotional pain, right? And so it's like, man, this is complicated, right? I mean, we have a lot of evidence to suggest that most mammals are going to respond physiologically to emotional and physical pain in a very similar way that humans would. And so what this forces us to do is to determine whether what we're doing is important enough to what's the term? I'm struggling to even remember the term. To justify what we're doing. And so, there are various, issues that you deal with regards to this. And so what it requires is what we call an institutional animal care and use committee, where typically you have people from the university in various places. You have administrators, faculty people, staff people, and then you have some people from outside of the university that serve on these university animal care and use committees and review every proposed research project, and to decide whether the benefit of that research outweighs the cost that is going on subjecting animals to what we would call or abnormal harm, right? Abnormal harm. All right. It would be harm that isn't associated with the normal way in which the animal lives, right? So if you're doing a study and you want to know, will this predator use this food source, right? That's not necessarily abnormal harm, right? Wild animals get eaten all the time, right? Removing tissue samples from a live animal and then stitching it up and allowing it to live for a little bit long, that's abnormal harm, right? That's not something an animal is expected to experience during its life. Okay. It's easier to get tissues from animals than it is from humans, especially considering that most of our animal models, we're dealing with really small organisms that develop very quickly. Okay? So you want to get an adult liver sample. With a mouse, you can get that in less than 3 weeks from birth. You can get an adult liver sample. Okay? It's a little bit more complicated than that with humans. It takes a little bit longer. And so it's just easier to get a lot of these tissues. Plus, if you're dealing with a small animal, a necropsy tends to take a lot less time and require a lot less resources than doing an autopsy on a human cadaver. All right, and then the last one is we can develop genetically modified animals. Now we can do this with humans. It's not a matter of what are we capable of technologically. Right? If you can genetically modify a mouse, you can genetically modify a human. The thing is there are laws against genetically modifying humans. There are not really as many laws against genetically modifying mice or genetically modifying fish. I mean, heck, you go to a pet store and you'll see all sorts of genetically modified fish glowing, right? These are genetically modified fish. I mean, if in about 5 minutes you can order 8 different types of genetically modified Drosophila and they'll be shipped right to your door, right? And so there aren't as many laws against it, which makes it basically like not only can we, use selective breeding to get a mouse that's going to develop type 2 diabetes even if you don't feed it ice cream every day, but you can get a mouse that's going to express a human protein, which no mouse would ever be able to do. But you can genetically modify it so that your mouse can actually make a human version of a particular protein, and not just a mouse version.
Are there some countries where they legally allow the genetic modification of humans?
That's an excellent question. I'm sure that there are. I would say that there are probably many countries that have no law against it, but most of the countries where the technologies to do so are readily available. Have some kind of a law in place. And then there are international laws against it, but how international laws go. It really just depends on what the country thinks about international laws, right?
Nazi Germany did experiments on humans.
Yep, absolutely. And, anyways, I was going to say something that was marginally politically charged, so I'll avoid that, so. Well, we can have an individual conversation if you want on how you should view international law, but it's maybe an individual type of conversation and not a group conversation. All right? So, any questions about this? This basically concluded what chapter 10 from our textbook addressed, and then so from this point, I need to demonstrate to you the challenges associated with using statistical models in comparative studies, and just how it's a completely different experience than when you're doing controlled experiments. controlled experiences. What does that even mean? Okay. All right. So, how are we going to do this? Should I keep it recording and then so you can still hear my voice but you won't see anything on the board? Okay, we'll do it. Do it. Okay, let's turn this off. That way we don't have to worry about it overheating. And if it gets loud outside, we can just close the door. Okay. So I want to show you, first, I'm going to demonstrate to you the two most common statistical tools that we'll use in genetics research when we're doing controlled experiments. Okay? The two most common tools that we'll use in controlled experiments in genetic research. First one is chi-squared analysis. And then the next one is basically something along the lines of this. A two-sample t-test. Some form of that. Okay, so these are our two probably most common statistical tools that we'll use in controlled experiments on genetics research. Okay, so first, the chi-squared analysis. Now, for those of you that have taken statistics at some point in your life, you know that there are several different forms of a chi-squared analysis. Okay, they come in several different flavors. Okay? So the most common one that we use is called the chi-squared goodness of fit model. And basically it works like this. It basically works like this. So what we're doing here is we're basically saying, okay, if these two genes were assorting independently, okay, if these two genes were assorting independently, we would expect these phenotypic ratios. But if they aren't assorting independently, or there's some epistatic effect between the two genes, so that the expression of one is influencing the expression of the other, we might get something different. Okay? We might get something different. So here's kind of what this would look like. I want to give you a realistic test. Okay? I want to give you a realistic test. So we're going to take a female, and we're going to take a female that, I don't know, let's say this female is homozygous dominant at one locus and homozygous dominant at the other locus.
That looks like something.
Yeah, let's do this. Let's do this. Okay, and it really doesn't matter what these are representing. We're just like, okay, these are two separate loci. These are two separate loci, and we don't really need to be concerned with what characters are actually influenced by this. And we're going to cross this female with a male that is homozygous recessive at that first locus, and happens to be heterozygous at the second locus. And we're basically living in a reality where we can form an infinite number of offspring. Okay? We're in a reality where we can form an infinite number of offspring. Don't ask me how that's possible, but that's how this works. Yeah, David.
The female, is it heterozygous on the Ys?
No, she's homozygous dominant there as well.
Okay, sorry.
Yeah, I know, it did look different. You're absolutely right. Okay, so we're going to do this cross. This female can only make one type of gamete, right? She can only make a gamete that has the dominant X and the dominant Y, because that's all she has. That's all she has. Okay, she can only make one type of gamete. This male, on the other hand, he can make two different types of gametes, right? He can make gametes that have the recessive X and the dominant Y, and he can make gametes that have the recessive X and the recessive Y. And we're like, cool. Okay, two different gametes that this male can make, and now we're going to cross these individuals. And so our F1 generation, the cross between this female and this male, basically has two different outcomes. Okay, since a female only has one type of gamete she can make, we only need to represent that condition. I mean, if you want, you could do like you typically would with a cross of two characters and do a Punnett square that has 16, 16 cells, but I mean, you're going to come to the same thing. All right, and so here's how this is going to work. So we'll do this cross. If this sperm fertilized this ovum, you would have this. And if this sperm fertilized this ovum, you would have this. And you're like, cool, Dr. Engel, that's really cool. But all of our offspring, and we can form an infinite number of offspring, they're all going to be the same phenotypically, right? So we're like, well, yeah, I mean, we can go ahead and actually assess them and to see what their genotype is, but they're all going to look the same phenotypically. But F2, this is where it's going to start to get interesting. So in F2, we basically, let's say we cross a random female from this, okay, and a random male from F1. Okay? Now a random female from F1, all of the individuals are going to be hybrids at this one locus. All of them are going to be hybrids here because that's the only option genotypically. But an average female, half of them, basically an average female has a 50% chance of being homozygous dominant and a 50% chance of being a dihybrid. Okay? Does that make sense? All right. But we're just going to focus on this dihybrid because I don't care about this one for right now. Okay? So we just executed this, and even though there was only a 50% chance, we lucked out on both our male and our female. Okay? So our female, for F2, we're going to cross a female that is a dihybrid with a male that is a dihybrid. Okay? And I want to show you what their chromosomes are going to look like. Okay? What their chromosomes are going to look like. Now, we know that we could have gotten a dominant Y from either mom or dad. Yes? But we didn't. Okay? So not only did we luck out and get a dihybrid female and a dihybrid male, but we also got a dihybrid female where both the X and the Y, or the capital X and capital Y, the dominant conditions, are on the same X chromosome, or on the same chromosome. I mean, it could be the X, it doesn't really matter. I saw X and I said X. Okay? And then here, we've got that, so it just happens to be on the same chromosome. I mean, this is just wonderful. Same thing here. And you're like, lowercase and lowercase. So this is lowercase. Let me draw that so it's clear. Okay? So we've got the one that came from, they got a chromosome from mom that had the dominant X, dominant Y. Their chromosome from dad has recessive X, recessive Y. Same thing for the male. Okay? We just lucked out because this is beautiful. And I need this illustration to work out nicely. Now, this female can make 4 different types of gametes. Yes? Assuming they sort independently. Right? Assuming they sort independently, this female can make 4 different types of gametes. Capital X, lowercase y, capital X, capital Y, lowercase x, capital Y, lowercase x, lowercase y. Right? Assuming they sort independently. Same thing for our male. 4 different gametes. Capital X, lowercase y, capital X, capital Y, lowercase x, capital Y, lowercase x, lowercase y. Okay? 4 different gametes. Okay, so now we need to cross these. I'm going to need room over here. Because now we actually are going to have to do a fairly complicated Punnett square. Okay, so we've got, I'll put them in order because you may have not liked the order that I use, so I'll do an order that I think you'll like better. You like that order better? That's conventionally how we tend to order it. Like, why do you care about convention? You do what you want, Dr. Engel. Should. Okay, so we already know this. We've done a dihybrid cross before. If you want, we can go and we can put all these genotypes. But we know that our phenotype ratios when we do a dihybrid cross, our phenotypic ratios should be what? 9 to 3 to 3 to 1. 9 to 3 to 3 to 1. Okay? 9 to 3 to 3 to 1. And this 9 represents dominant for both. So I'm going to say dominant for both. Yes? Okay, that's what the 9 represents. That they've got the dominant phenotype for both. For this one is dominant for one and recessive for the other. Okay, and that's true for both of these. It's just the other way. And then this 1 is recessive for both. Okay, so this is what and again, we have the ability here to have an infinite number of offspring. And so we're in no way concerned that our offspring ratios are some statistical anomaly. Okay? We okay so far? Alright. Do you love it? Like Dr. Ingo, I love this. May I erase this? Okay. I heard yes, nobody said no, so it's going. Okay. So that's what our phenotypic ratio should be if those two loci are sorting independently. And the beauty of this is it doesn't really matter whether it's an independent assortment issue or whether it's an epigenetic issue. We're really looking at is one locus influencing the other? Is it influencing the other because they're too close to assort independently, or is it influencing the other because its expression might mask the expression of another? We really don't care. All we really care is about the phenotype, and does that match what we would expect? Because if it doesn't, it means that these two loci are related in some way. They're related epistatically, or they're related based on location. We okay with that? Okay. So this is what our ratio should be. Now I'm going to do some simple math here. I'm going to make the math simple. And let's say we took a sample of our infinite number of offspring. Okay, we took a sample that happened to be 100 or 1,600 offspring. Okay, we just took a sample, and we're pretty confident that this sample represents all of our infinite number of offspring. Okay, now what we would expect. So now here we're going to build a little chart. And this chart, I mean here, this will be confusing. Let's just make this one row. I don't really like one row charts, but whatever. We're going to do that. So our expected values, if these two genes were not related in any way, of these 1,600 offspring, 900 of them would be double dominant. Dominant for both, right? 300 of them would be dominant at X and recessive at Y. 300 of them would be dominant at Y and recessive at X. And 100 of them would be recessive at both X and Y. Okay? Those are our expected values. Does that make sense? But that's not what we got. Because when we sampled these 1,600 offspring, that again, we are confident they represent our infinite offspring produced, this is what we observed. Let's do this. 1,100. Okay. And then, I don't know. We'll keep this one 300, but then this one will be 100, and this one will be 100. And I don't even think that adds up. Does that add up? Oh, it does. It adds up to 1,600. Excellent. I think so. And it's not Monday, and my math usually is pretty good outside of Mondays. Okay? So this is what we observed. And the question is, are they actually related? Are they too close to assort independently, or is there some kind of epistatic relationship? Well, we have a chi-squared goodness of fit test that will allow us to have a statistical tool in which we can say, are these data different enough that they aren't the same, that they're really, these genes are really linked in some way, either epistatically or by location. Okay? And so we use what's called our chi-squared, and what it equals is this. It equals the sum of our observed minus expected quantity squared divided by the expected value. The sum of our observed minus expected squared divided by the expected value. Right? That's so cool. And so what this means is then what we need to do is we need to take our observed minus our expected. So 1100 minus our 900. Okay? Right? And then we square that, and then we divide that by our expected, which is 900. And then we need to add that to the next one, which is going to be 300 minus 300 squared divided by 300. This one's just going to equal 0. We can just eliminate that right away. And then we need to add that to 100 minus 300 squared divided by 300. Right? You okay so far? Mm-hmm. And then we need to add in the 100 minus 100 squared divided by 100, and that one's just going to equal 0 as well. So we don't even need to worry about that one. Okay? We okay so far? Okay. And then, then we do, 1100 minus 900 is 200. 200 squared would be 40,000. Yep, 40,000 divided by 900. Man, if it was 40,000 divided by 1,000, it'd be 40, but divided by 900, it's like, what is it, 54? Anyone do this? Now I'm going to have to actually do this.
44.4.
44?
You said
40,000 divided, oh yeah, it is. Man, that was nowhere close. 44 plus, and then this one is going to be 200, or negative 200 squared, which, is just, the same as doing 200 squared, so that's going to be 40,000, but now it's going to be divided by 300, which is going to be plus 133, and we get that our chi-squared value is 177.
Square.
What's that?
Is it equals x to the square power c?
Oh no, it's just chi-squared. It's a Greek letter, and the chi-squared is the entire variable. Yes. Okay, so our chi-squared value is 177. So then you can go and look at a chart and find your critical chi-squared value, and your critical chi-squared value is going to be for these few data probably 0.5. That if your chi-squared value is anything bigger than 0.5, you're like, wow, these genes are really linked in some way. And our chi-squared value, it's huge. It's huge. And so what's happening is we're getting way more of the parental chromosome coming along than really what we should. And so they're not assorting independently. They're either not assorting independently because they're too close together, or there's some kind of epistatic effect that's producing a phenotype at an unusually high ratio. That this phenotype is appearing way too often, and this phenotype is appearing way too little.
So you said if it's over 0.5, then they aren't linked.
Yeah, I don't know what the chi-squared, because this only has 3. Yeah, I mean it has 3 degrees of freedom. So I'm just guessing that a chi-squared value. I mean, if you want to go through, you can actually figure out what the p-value is here, or you can just trust that this is a huge chi-squared value. Okay, and so basically what we would do here, it's not all that complicated of a statistical tool. I mean, yeah, it took us a little bit of time. We had to do some math, but it's like observed minus expected squared divided by expected. Like I can do that, right? It's not complex mathematically. It's just I need to know what chi-squared is and how to solve it. And that's about as complicated as it gets, because doing a 2-sample t-test is super simple. You have an average of one group, you have an average of the other group, and then you compare those two averages. Well, you also have to take into account, how much variation there is. But I won't even walk you through this because nobody does this by hand. You would just plug in your data and it's very simple. And it tells you basically what's the probability of getting averages that different if the two groups are really the same, right? And so this is where you would do this is like you take this population or you take this group where all of them have the genetic illness, you take this other group where none of them have the genetic illness, and then you're basically like, what proportion of people in these groups have this specific mutation, or this specific combination of mutations. Okay, that one's very simple. Chi-squared's probably the most complicated statistical tool that we would ever do in a controlled experiment on one of these genetic studies. Yeah, John.
Wait, so what's the relevance of the 177?
Well, so the relevance of the 177, you can basically answer that question in two ways. One, it's you can determine what chi-squared value do you need to hit to where you have your probability of making a type 1 error is less than 5%, which is typically where we set that limit, right? When our probability of making a type 1 error is less than 5%, we're like, these are really significant differences. So you can look at it that way and just compare this to what you would call your critical chi-squared value. When you have these few of data, what does your chi-squared value need to be in order to hit that probability of making a type 1 error?
How would you find that?
You just look it up.
Oh.
Here, let's look it up right now. I've got my computer.
So you say chi-squared standard for 1600, like a sample size of 1600?
Oh no, it'd be based on these number of data for basically four different groups. So it wouldn't be of the 1600. It would be, let's see, critical, well, that's caps lock, not A, critical chi-squared value with 3 degrees of freedom. It's actually 7.8. A chi-squared, it's not 0.5. The critical chi-squared value is 7.82 when you have 3 degrees of freedom. What's that?
It was 7.8.
It's 7.8. Yeah, is that what you were guessing?
I guess 8.
Did you? 7.82 with 3 degrees of freedom. So it would give us if you have a chi-squared value that big or larger, you have a 5% chance or less of making a type 1 error, of saying they're different when they're really not. And so our chi-squared value is greater than that, which means our probability of making a type 1 error in this situation is significantly lower than 0.05. So for instance,
So
if
our error is just saying that our error would be saying that these two numbers are
saying in this context, the error, your type 1 error, would be saying that these two loci are linked in some way when they're really not. Right? Saying they're linked epistatically or saying they're linked because they're really close together on the same chromosome when they're really not. Okay? So let me give you some context. If your chi-squared value were 16.3, you would have a 1 in 1,000 chance of making a type 1 error. But our chi-squared value is 177. So our probability of making a type 1 error in there is astronomically small. Yeah. Okay? Alright. So that's one way you can do it, or you can just go and you can take this specific chi-squared value with the specific amount of degrees of freedom you have from the number of basically observations, and we only have 4 observations. There's a count for each one, but it's 4 observations, 4 phenotypes. And so you could take your chi-squared value with your specific degree of freedom, which in this case would be 3, and if you want to know why that is, we can talk about that. And then figure out what your probability of making a type 1 error is.
okay.
Yeah, but again, that's just something you look up. Now, no chart is going to give you this, so you'd have to use some kind of a software that would actually look it up. But our probability of making a type 1 error in this situation with a chi-squared value this high is going to be astronomically small. Maybe 1 in a billion, 1 in 50 billion, I don't even know, but it's going to be really small.
May I ask what the degrees of freedom are coming from?
Yeah, so for these chi-squared goodness of fits tests, your degrees of freedom is basically a representation of it's acknowledging the fact that the more observations you're making, the more opportunity there is for error, right? So you need a larger chi-squared value before you're really confident that your differences are really legitimate and are not just due to chance, right? So what you would typically do is you would do the number of rows minus 1 times the number of columns minus 1. But we only have 1 row, so 1 minus 1 is 0, but this is not 0 degrees of freedom. Okay, so what I was doing here is just providing an estimate of our degrees of freedom, and I was providing a very conservative estimate. But if we represented these instead of a table with 1 row and 4 columns, if we represented it like this, 2 rows and 2 columns, we'd still have 4 observations, right? And then we would have 2 rows minus 1 is 1, 2 columns minus 1 is 1, 1 times 1 is 1 degree of freedom. Okay, and so, I mean, here, I'll tell you what but I was being conservative with 3 degrees of freedom. It's probably closer to 1 degree of freedom. And let's see. Critical chi-squared value with 1 degree of freedom, 3.84. A chi-squared value of 3.84 would give you a probability of making a type 1 error of 0.5, 5%. 5% chance of making a type 1 error. All right? Okay. Thoughts, concerns? Okay. Now I just want to show you some of the challenges with comparative studies, and we have like 6 minutes. I don't know how much I can accomplish in 6 minutes. Okay.
Did you not erase all of it?
Oh, not erase all of it? Oh, okay.
Just that little corner.
Yeah. Fine. So when you're doing a comparative study, you really have 2 legitimate options for determining your correlation. You have 2 legitimate options, okay? I mean, in most cases. You have what's called a linear regression. And you have what's called a logarithmic regression. This is what we have when we are doing comparative studies. These are going to be our 2 most helpful tools. The linear regression is really nice. It's basically asking, you know, if there's a linear relationship or an approximately linear relationship, as your independent variable goes up, so does your dependent variable. So in this context would be as your frequency of a particular mutation goes up, does the incidence or prevalence of this particular genetic illness go up? Right? Does that make sense? So if we're doing, let me show you a picture of a linear regression. One in which you would actually have a really strong linear relationship. So on the x-axis here, we have proportion of mutant allele. And over on the y-axis, we would have prevalence of illness. Do those variables make sense? Okay, proportion of mutant allele, and then on the y-axis, prevalence of the illness. So what we would do is we'd study several different groups of people. Like maybe we just sample people from this family, and we'd say, okay, 10% of the people in this family have this specific mutant allele. And this percentage of people in that family have the genetic illness. And then we study a different family, and it's like, okay, 20% of the people in this family have this particular mutant allele. And this percentage have the genetic illness. So this would be a very strong linear relationship here. And so what this is saying is that as your proportion of this specific mutant allele increases, so does the prevalence of illness. And this would demonstrate that there is a very strong relationship between this specific mutation and the prevalence of this genetic illness. Okay? If you had something like this, you would be like, we are very confident that this locus with this specific mutant allele is related to the presence of this genetic illness. Okay? And we're like, cool, that's just wonderful. But this is, again, this is a little bit more complicated to do. The analyses are a little bit more complicated. How you interpret these are a little bit more complicated because you're not really concerned as much with what your curve looks like as much as you are with what's called your R-squared value. So your R-squared value basically equals this. It's the percentage of variation in y, and this is the variable on our y-axis, percentage of the variation in y explained by variation in x. And that's really what we're concerned with. And this is how we do some of these tests to approximate the heritability of traits. When we're like, you know, the heritability of human height is approximately 80%. What we mean by that is 80% of the difference in human height can be explained by differences in the genetics of people surveyed. Okay? That's what we mean by that. Now, what this requires, in order to do a linear regression, you must have a continuous dependent variable. In order to even do a linear regression, your dependent variable has to be continuous, which means that any value needs to be possible. But here's the issue. Can you have a negative prevalence of genetic illness? Prevalence is basically what percentage of the group has that genetic illness. Can you have a negative percentage? You can't, which means you really can't even use a linear regression. I mean, people still use it, but if somebody really got upset about it and one of your reviewers on your paper put up a challenge of like, hey, if not all values are possible, you can't really use a linear regression. Which means you're left with this, a logistic regression. And a logistic regression, or logarithmic, is basically, your dependent variable might as well just be yes or no. Dependent variable should just be just narrow it down to yes or no. Does the person have the genetic illness? Yes or no, and then their particular genotype or their particular combination of single nucleotide polymorphisms. Because once you can't really do this, which people still will do this, even though not all values are possible and it's not truly continuous, as long as it's continuous between your minimum value and your maximum value, you sometimes can get away with it, with doing a linear regression. And so if any value is technically possible between 0% and 100%, you might be able to get away with it. But it's a lot more complicated, and we can't really say anything about cause and effect. It's all about correlation. It's all about what percentage of the variation in y can we explain with the variation in x. All right, we are out of time. You should be fully equipped